Today's webinar is going to be on the European Nucleotide Archive, and we're joined today by Sam Holt. I'll just pass over to Sam now. Okay, thank you.
Okay, thank you very much, Anna. So yeah, I'm Sam Holt, and I'm the user support bioinformatician at the European Nucleotide Archive. And today I'll just be running you through some basics about us. So hopefully by the end of this, you'll be a bit more familiar with who we are and what we do, and you'll know what services we can provide, and you'll know how to go about accessing them, and they'll seem familiar to you so that you know where to go to look for help when you do come to use them. So here's how the talk will break down today. So I'll start with just quite a general introduction of who we are, what we do, and why we do it. And then we'll go on to talk about the data and metadata model, which might sound a bit impenetrable, but that's just how we organize data and the information about that data to make it useful to people such as yourselves. And then we'll go into the practical topics. We'll talk first about how you can find and retrieve data. And finally, we'll look at how and why you can submit your data. So, what does ENA do? We provide a comprehensive record of the world's nucleotide data. So this means that huge amounts of nucleotide data are deposited within ENA, and we make it available to the public completely free and open to everybody. You can come and download the files. That includes DNA and RNA. So really then our purpose is to provide tools for submitting and retrieving data. That's really what we see as our function. I want to mention that we're the European node of the INSDC. That's the International Nucleotide Sequence Data Collaboration. And all that means is that we share data with two other databases. There's the NCBI databases in the US from GenBank, Sequence Read Archive, Trace Archive, and Bioprojects, and DDBJ, the DNA Data Bank of Japan. The three of us share data on a daily basis, and so anything that gets submitted to one of us gets submitted to the other two as well. Beyond that, we're very much linked in with other resources around EMBL-EBI and beyond. So what this means is that quite a few different groups take our data and subset it in some way, reanalyze it, present it in a way that is useful to a more specific community. And we do lots of data coordination projects ourselves. So we work with different groups and different projects to present data in a way that is useful to them. So for example, you can visit the FANG browser down here, the functional annotation of animal genomes, if you're interested in the genomes of agriculturally relevant animals. That's data subsetted from ENA, but represented in a way that is useful to that community. So as well as big collaborations like that, individual researchers, maybe such as yourselves, can benefit as well. And I want to highlight that with an example just for a moment. So what you're looking at here is a polyoma virus. Polyoma means many tumors. So it may be apparent to you, therefore, why polyoma viruses are not good. Prior to the work I'm going to discuss on the next slide, they were quite well known in mice and primates, including humans, but not really beyond that. And with viruses, it's really useful to have a good understanding of the taxonomy of a family, because that way when we discover new members of the family, we can do a quick bit of sequencing on them and quite quickly work out where they fit into their family, and therefore what we can expect of them and what sort of treatments might work. It's also just interesting. As scientists, we like taxonomy. So this is the sort of thing we want to know more about, but previously not many polyoma viruses were known. So addressing that was the goal of this particular paper, which would be published eventually under the name of Buck et al. Their paper described the discovery of new polyoma viruses in fish, cows, and sheep, and they determined that by doing some primary sequencing of their own from samples. So this is great. Previously, we only knew about polyoma viruses in terrestrial vertebrates, but now because we found them in fish, we know they exist in non-terrestrial vertebrates as well. But they went a step further and searched their sequences against INSDC data. And because they did that, they were able to identify likely new species of polyoma viruses within the genomes of 3 more vertebrates, 5 different spiders, and 2 different insects. They also found a near-complete polyomavirus genome within the genome of the Baja Californian bark scorpion. You may know that some viruses can embed their genome within the genome of their host as a survival mechanism, and sometimes that embedded genome becomes fixed within the host genome. And that's what happened with this bark scorpion. And Whoever sequenced this bug scorpion probably wasn't thinking about polyomaviruses. They might never have heard of polyomaviruses, but because they made their data available in a public archive, it was possible for this other group to come along and pull this completely original observation out of it. And so now we kind of have this evidence that the polyomavirus must have existed in the last common arthropod vertebrate ancestor. So now let's move on to talk about the data model. So this is the question of how we treat data and how we organize it, because we store a huge amount of data. And to give you an example of that, we, as of earlier in March, we contain more than 8.5 petabase pairs of raw read data. And this is data that's coming from many different users, they're sampling many different taxa, they're preparing their libraries using lots of different techniques, and then they're sequencing those on lots of different platforms. And we really need to have a consistent way to store and display that data. When someone comes to us with some data, we need to know right away what we're going to be doing with it. This is especially important because a lot of our submission systems are automated now, so we need to be able to direct those submissions without even any human intervention if possible. So the first step in achieving this is having a robust data model. And the way this works is that all sequence data is organized into one of three data tiers. So this is a tiered model, and at the bottom of it is the reads layer. So this is the raw output of sequencing machines. Typically, we think of this as FASTQ files. There are some other things, but this is the raw output of a sequencing machine with minimal processing done on it. Maybe some demultiplexing is done or some trimming of adapters, but that's what's in the reads layer. Built on top of that is the assemblies layer. So this is the result of using some kind of algorithm to re-examine those reads and line them up against an existing genome sequence or put together a new genome sequence. Either way, this is using some computational method to reconstruct those reads into something which represents a real biological molecule, like a chrome or a contig, which is a subsection of a chromosome. And the final tier is the annotation tier. So this is all about function. This is projecting some function onto that assembly, saying between coordinate X and coordinate Y, we have this coding sequence, for example. But that's really the data model. It gets subdivided a bit more, but that's basically how it works. All sequence data gets organized into these three data tiers. So here we see a FASTQ file, and this is an example of data from the read tier. But it's not very interesting because it's just a bunch of letters in a file. It doesn't tell us anything. We can't learn anything by looking at this file. To make this interesting, we need metadata. We need to know how that data was produced and why. So that brings us neatly to the metadata model. And this tells us all about how data was produced. So without any context, the data has no value. But when we have good metadata, when people submit good metadata, we know how sequence data was produced, and we can do interesting comparisons between data sets even. So have a look at this query here. I want to see data from bacteria in the Atlantic Ocean, sampled between 50 and 100 meters down, between April and July, compared with the same from the Indian Ocean. Those are two separate queries that you could run fairly easily at ENA, and you could look at the results, download some of the data, and do some interesting comparisons. This is the sort of thing that you can do because we have this robust data model. Briefly then, the way this works is that as well as submitting the data, you submit some other things, including runs, experiments, studies, and samples. And these are just descriptions of different aspects of your project. And to make this a bit more intuitive, I'm just going to talk through an example of how this might work using a real study that exists in INSDC. So what you're looking at here is a firefly squid. These exist around the Sea of Japan, and they're bioluminescent. They're very beautiful, and every spring they come to spawn in Toyama Bay in Japan, and they provide a really beautiful show of bioluminescence. They have these organs across their body, and that's what you're seeing in the picture here. They especially have these organs on their arm tips and around the mantle. But prior to the work that I'm going to be discussing today, it wasn't really known how this effect worked. It was known that it was produced by some protein crystals, but what those proteins were, how they worked, and what homologs might exist wasn't previously known. So this was really the goal of the study that I'm talking about here, to kind of establish what's already known and then to take it further by doing some sequencing to better understand these squid. So a moment ago we talked about the different metadata objects that get submitted, and one of these is a study. And what we're going to do as we talk here is build up a map of how this looks. So right now we have only submitted our study, and this has its own accession number and includes details about what work was done and why. And it's also going to serve as an umbrella to contain all the other things that are going to be submitted. So the first thing they do, having decided that they want to study these squid, is gather some samples of them. And that's not too hard because when they come to spawn, the fishermen get out and catch some. They're apparently very delicious. So you can just wander down to a fish market at the right time of year, and they're not hard to find in Japan. So they took 4 of these squid back to their lab, and they took samples from the arm tips and mantles, and they took multiple samples from different individuals because they're good scientists. And you can see here we have 4 of the arm tip samples and 2 of the mantle samples. And they log the details of these in the database, and this means that people who look at this in the future will know what organism the data were produced from. And so you can see our map now contains, as well as the study, 6 sample objects. And each of these samples is going to have its own accession number, and it contains details like the tissue that it came from and which squid it came from. I don't have much space on this slide, so I've included only minimal information. But of course, the real samples include information like the species scientific name, the geographic location that they were taken from, and the developmental stage of these squids. So having sampled these squids, the next thing to do is then to extract mRNA from the organs that they're interested in. The bioluminescent organs, because this is an RNA-seq study. They take mRNA, they make it into cDNA, and they sequence it on this machine, which is an Illumina HiSeq 2000. And so having done that, they then need to log the details once again of how they actually did the sequencing within the database. And so now we have 6 experiment objects. And you can see that these contain details of how the sequencing experiment was done, the fact that it was RNA-seq, the fact that it was a paired end experiment, and the machine that it was done on. The experiments are once again each going to have their own accession number, and they point to the study and sample that are relevant to them. So this is starting to introduce some connectivity into this model. The experiments are telling us information about what they're linked to. And the end result of having done all this sequencing work is FASTQ files. We did 6 paired end experiments here, so we end up with 12 FASTQ files. And so the researchers compress these, they upload them to the database, and they undergo some processing, which we do, and the files, once they've been accepted, are then there waiting to be made public. And what you're looking at here is a FASTQ file, and this is actually the same FASTQ file we looked at earlier, and hopefully you'll agree that it's a bit more interesting now that we've established some metadata facts about it. So we've submitted our runs now, and once again, each of these has their own accession number, and there's one pointing to each experiment that it was derived from. And the main thing that the run does other than that is point to the location of the files within a server here at EBI. So as you can see, each of these is describing the location of the 2 FASTQ files that it contains. So that's really all there is to the metadata model. Of course, you can complicate it a lot more than this, but this is a perfectly fine example of a study as it might be submitted to ENA. If this makes sense to you, then you're going to have a lot of luck using ENA. You'll find it quite easy to work with. So now that we've kind of established what ENA is and what our data is and how we store it, for the rest of this webinar, we'll be talking about the practical topics of data retrieval and data submission. So the point of having all this data is that people can come along and retrieve it. So how do they do that? Well, if you were to go to the ENA website right now, one of the first things you would see is a search box like this one here in the top left of the screen. And you could put something in there and you could look for data. So in this case, I've put human kinase in there because I'm interested in finding human kinase data. And you can see the results that I got at the bottom here. And you can see we got results from all kinds of different domains. You can see we got, for example, sample, study, and read results. These are the things we were just talking about. We also got results from these different annotation domains. And that's what we were looking for. We were looking for annotation data. The fact that it's a kinase is functional information, so this is annotation data. And if you look at the results, you can see, for example, that one of them here is human protein kinase mRNA. So that's great. That's the kind of thing we were looking for. But if you look further down, you'll see that this next result here from the coding section is human gamma herpes virus kinase. So this is not from human, it's from a virus that infects humans. So you can see right away that the simple text search here is quite prone to false positives. So it's perfectly good if you already know what you're looking for. If you put an accession number in here, for example, this will lead you right to it, but it's not the most intelligent way to run your searches. The best thing to do is to use the advanced search browser. So when you come to our advanced search page, the first thing you'll see is this domain selection thing. So this allows you to specify what sort of thing you're going to look for. And once again, there's some things which by now might be familiar. You can search for samples, you can search for studies or reads. In this case, we've searched for sequence because we're interested in annotated data. And when you click that, the first thing that happens is more boxes appear beneath. And we've got the taxon box here, where we can specify what taxon we're interested in. Now we're interested in human. When you start typing into this box, you see suggestions appear. And that's because whatever you put into this box has to match with something in a taxonomy database. And the advantage of this is that it means that whoever submitted the data that you turn up submitted it saying positively that this belongs to this taxon. So when you put human into this box, anything that turns up will be something that somebody has positively identified as originating in a human. It works with synonyms too, so it doesn't matter if you put human or Homo sapiens in here, you're going to get the same results there. And having restricted the taxon, we're now going to restrict the description. We're going to put kinase in here, and we're going to surround it with these wildcard symbols, which just mean anything. So this is going to restrict our results to anything that contains kinase in its description. So what we've done here is the same search, but we've focused it on a specific type of data. We've restricted the taxon, and we've restricted it, the description that we're going to allow. We're going to leverage the fields that people submitted when they submitted their data to find a much more relevant data set. And the results look like this. And again, you can see down here, we only have one data type now. And you'll just have to trust me that we have a much more limited and restricted and relevant set of results here. And we can click through them to find sequences of interest. So restricting our search gets us much more relevant results. And you can focus your search on all kinds of things. Here we focused it on taxonomy, but you could also focus it on geography. For example, to find the squid samples that we looked at a minute ago. You can focus on environmental conditions, experimental protocol. There are a lot of different things you can focus on. So for the last little bit then, let's talk about how you actually go about submitting data. So we're going to start then by talking about why you would submit data, because all our data comes from members of the research community. We don't produce it ourselves, it comes from people like you. So what motivates people to submit? Some people are submitting for fairly high-minded reasons. For open data reasons, we believe that data should be freely available so that the claims that people make in their scientific papers are reproducible, and there's a trail of evidence that allows them to support their claims. There are also more practical reasons why it's useful. Uploading your data to a public archive makes it accessible to third parties, so it can be helpful to share with perhaps collaborators who are geographically remote from you. It's useful for archival. If you're a relatively small sequencing center, then the overheads of storing lots of bulky sequence files can become quite a problem. But if you submit it to a database like ENA, then it is going to be archived in perpetuity, and you don't necessarily need to keep your own local copies. A lot of journals will require that your data has been made public before they will allow you to publish with them. So you may have to do it to satisfy the requirements of publishing an article. And finally, there are groups like Magnify. Magnify are a group here at EBI, and they work with metagenomics. And if you upload metagenomic data to ENA, for example, then you can get some free analysis done on it by Magnify. They'll give you some information about like the taxonomic makeup of the community that you've sequenced. So there are these kind of practical analysis benefits that you can get out of submitting data as well. So when it comes to actually submitting the data, there are 3 different submission routes, and the reason for that is kind of historical. They're all good for different things and can submit different things. So we'll talk a little bit about that as we go. But broadly then, there's the interactive submission route, and this is where you use your web browser to fill out forms that describe your work. There's Web-in-Cli. This is a command line submission interface that we have put out in the last year. And there's the programmatic submission route, and this is the older route that kind of underpins all the others, and it involves describing your work in XML format and submitting it to us using an HTTPS protocol. So the interactive route then, this, as I said, allows you to register your data by just filling out forms in your browser. So this is what's most familiar to most people. It's quite accessible to use. It doesn't require any particular specialist knowledge. For larger submissions, you can prepare a spreadsheet and submit that through this interface, but that's really about as complicated as it's going to get. And I've included a couple of screenshots from this interface here. In this case, we are submitting some samples. So remember that samples are the kind of biological origin of the data that you have produced. And when you submit samples, you have to fill out a checklist. And here you can see some of the environmental checklists that we offer. So you could, for example, select the human gut checklist, and this would suggest that you had sampled human gut metagenomes. And when you select one, a specific set of boxes appear that describe a human gut, and there are different requirements and different directions on what you should submit. But that's the interactive route. It's quite familiar to most people. So Web-in-Cli, as I said, is a particular tool that we put out in the last year. It works from your computer to validate your files before you submit them. So it will tell you whether they are going to be accepted before you actually get to submit them. And it gives you a chance to correct any errors that are wrong with them. So once you've prepared your files, all you have to do to use Web-in-Cli is prepare a manifest file, and that is this thing over here on the right. And this is just a list of values, and you can see on the left what is being described, and on the right what the value is. So here we have some read data that we're submitting, and we've named our library reads1. We are stating that the platform that we did the sequencing on was Illumina. We are saying the insert size was 200. We are specifying sample and study accessions, which we want our read data to belong to, and we're specifying the location of the FASTQ file, reads1.fastq. So having prepared a manifest file, all you have to do is put together a short command line, and we have guidance on how to do this, which I'll link you to at the end. You invoke the program by typing its name. You tell us what you're submitting, in this case reads. You tell it where to find the manifest file, lib1manifest.txt, which is this thing. You tell it that you want to submit, and then you give it your login data. And that's all there is to using Web-in-Cli. It will spit out the results of the validation for you and give you a chance to correct any mistakes in your submission. So the final thing that we have to talk about then is the programmatic route. So this is the third submission route. And as I said before, you prepare an XML file describing the submission. An XML file looks a bit like this. Unless you're doing very large submissions, or you're running some kind of sequencing center, or you want to submit one of a few quite obscure data types, you're unlikely to have to use this. But I just want to make you aware of it so that if it is something that might be relevant to you, then you know it's out there and you can come and find more information about it. So here we have a sample object described in XML format, and you can see we just have these tag value pairs here. So for example, we can see that the age of the organism here is 15.5. We can see that it was collected on the 25th of March 2016 at 10 a.m. And that's all the XML file is doing here. And you send it to us using a different command line program. Most people use this one called curl. where once again you just give your username and password, you tell it where to find the files that you want to submit, and then you specify the URL that you want to submit to. So this is our address here for ENA. This is where you would submit to. But that's really all I want to talk to you about for this afternoon. Hopefully now you have a clearer idea of what services we can offer you, and the main thing I want to leave you with really is this link here. This is the link to our submission tutorials. So this is where you can get advice and practical help on how to actually submit your data. And if that doesn't prove to be enough, then we operate a help desk, and that is most of my job. And the address for that is datasubs@ebi.ac.uk. So we'll take any questions now, but if you have any later that you forget to ask, or you have problems submitting or retrieving data, then this is where you can go to get help. Read the docs page here and the email address here. So thank you very much for your attention.
We've just one final slide. So if you would like to watch some more of our webinars, the list of upcoming webinars will be on the link just on that slide. Please go and have a look and see what we've got coming up. Okay, thank you very much for listening. We'll leave there. Again, if you have any questions, do email afterwards. But that'll be all for this afternoon's webinar. Thank you.